and welcome to Intro 101. My name is Chris and I am your neuroscience and psychology graduate student assistant. I'm here to assist you with the related science basics that you interact with on a daily basis. My goal for doing this is to help you start or continue to build a foundation in human biology and physiology that can better help you engage with the information you consume from your favorite health and wellness educators and influencers, much like the way a graduate assistant helps prepare intro students so they can better engage with lectures delivered by their professors. Or maybe you're here today because you'd just like to know the biological and physiological precursors to the main event that launched the very beginning of you. Either way, stick with me for the next many minutes, and I'll do my best to explain the physiological prequel to how you began. Today's episode is Intro to Neurodevelopment 101, Part 1, Gametogenesis. It is entirely devoted to what was happening inside your biological parents' reproductive systems that made it possible to create you. So you may recognize that the reproductive system is not directly part of neurodevelopment per se, but aspects of reproductive processes can have downstream effects on genetic, molecular, and cellular components of nervous system development. Over the next few episodes, we're going to follow the processes of neurodevelopment from the pre-fertilization stages. We'll call that the prequel to the main story. And then part 2 is the physiology of fertilization. Part 3 is implantation, gastrulation, neurulation, and neurogenesis. This is where neurodevelopment really kicks off. But as I said, reproductive processes leading up to it can influence its development. And then in part 4, we'll get to infant, childhood, adolescent, and young adult neurodevelopment. It's a lot to pack into only a few episodes, so I will be sticking as closely to basic biology, physiology, and some homeostatic processes as possible. Though we will look at what can happen when these different stages of development veer away from textbook typical developmental trajectories, and how that can potentially lead to atypical outcomes. There are a lot of factors that influence every part of human development, from the state of sperm and egg cell health prior to conception to genetic and environmental impact on fetal development. And then it gets exponentially more complex once we are born into the world. So I'll say it now, and probably a million more times over the next few episodes. We don't yet fully understand the etiology of many atypical outcomes or all of the biological markers involved in infertility, fetal development complications, or neurodevelopmental disorders, including those that lead to neurodiversity. There is even debate about what neurotypical actually means, given the incredible diverse developmental variability among any population of people. And do we actually have adequate representational comparison models in our current gold standard assessment toolkits? So please, do not use the information presented in the next few episodes to self or other diagnose. For this particular episode, if you have concerns regarding fertility, please speak with a licensed medical professional or a family planning organization with licensed medical oversight that can provide resources or assessments. I'll post any helpful links I know of in the show notes, but if you are aware of any others, do let us know in the comments below. Before we start, I'd like to respectfully acknowledge that Intro 101 is recorded on the unceded and ancestral territories of the Kwantlen, Katzie, Matsqui, and Semiahmoo First Nations. Additionally, we at Intro 101 believe wholeheartedly that science is for everybody. In fact, the richness, complexity, and strength of scientific research is astronomically improved by the inclusion of many varied voices. To ensure everybody has access to education, research resources, and representation across scientific fields the world over, I will be sharing organizations in the show notes that provides access to STEM education and research resources, and ask that you check them out and support them in any way that you can. If you already advocate or generate opportunities to provide equitable access to STEM education and resources within your own communities, or support other STEM education organizations not listed here, please share them in the comments below if you'd like others to contribute as they are able. All right, welcome to class. Thanks for being here. Let's crack on with Intro to Neurodevelopment 101, Part 1, the prequel, Gametogenesis. First, I'd like to mention, if you've just arrived at this channel through this episode, this series is an episodic integration of information that is building a bigger picture of the nervous system, its components, physiology, and its collaboration with other body systems to keep your body in homeostasis. If you're new to homeostasis or the nervous system, or a little rusty on how it works, it may benefit you to view the previous episodes in this series first. A general understanding on what homeostasis is, nervous system function, and general cell physiology and function will help make sense of this episode, all of which are covered in the first 3 episodes in this series. Now, where to begin? I was originally going to launch directly into human egg cell fertilization and what transpires from that magical moment onward, but then I thought it might be helpful to rewind a little bit and give some context to the physiological and regulatory reproductive mechanisms involved in sperm and egg cell production, which can be helpful in understanding the underpinnings of fertility, even if you're not planning to start a family or struggling with infertility. It's also helpful if you'd like a better understanding of your own reproductive health and function, or that of your partner if they're of the opposite sex. Also, basic understanding of reproductive physiology can be a helpful primer if you're interacting with health and wellness influencers and educators who deal with reproductive health. That said, I also think it's important to convey that this episode may contain triggering language for those who have experienced or are experiencing fertility issues. If that describes you and you wish to proceed watching or listening to this episode, I recommend having a contact list that includes safe people, organizations, or a therapist you can reach out to in the event the information in this episode triggers emotional overwhelm. There's nothing intentionally upsetting about this information, but with any difficult experience that affects our emotional and mental health, it can be surprising what can indeed affect us. And what affects us one day may not affect us the next, and vice versa. So it may benefit you to have a contact list or autonomic regulation plan going into potentially triggering territory. For everybody, there is a somewhat graphic description of what happens to a uterus during menstruation. Consider this fair warning. Also, a point of admin to mention up front. I fully recognize that there are people whose reproductive organs do not align with their experienced gender, and I will do my best to use inclusive language. Okay, so, gametes. What are they? Well, the bare basics of growing a human typically begins with the union of gametes. Gametes are reproductive cells, and in mammals, those are sperm cells, or spermatozoa, and egg cells, also called oocytes. Matured oocytes are called ova, but they don't technically fully mature until after fertilization, so I'll be referring to egg cells as oocytes for most of this episode. Anyhow, these gametes, or reproductive cells, are produced in the gonads. Gonads are reproductive organs, and in humans, the gonads will either be a pair of testes or a pair of ovaries. On average, human males have testes and human females have ovaries. This is not always the case due to myriad developmental outcomes. There are a number of developmental anomalies that can interfere with gonadal development. For instance, gonadal agenesis and dysgenesis and their subtypes result in either arrested gonad development or improper gonad development. This is noteworthy because gonads have 2 basic functions. One is to produce gametes, but the other is to produce and secrete steroid hormones that help regulate gamete maturation and release, among other hormonal regulatory functions. Without functioning gonads, steroid hormone production, like testosterone and estrogen, may be limited or non-existent and may require lifelong hormone therapy as early as pre-adolescence to induce puberty, as well as contribute to other essential physiological outcomes. An important note about gametes that is different from typical cells is that both egg cells and sperm cells only have half of the usual 46 chromosomes among their genetic material. It's a little more complicated with egg cells, but generally speaking, after the egg cell and sperm cell unite, the 2 chromosomal halves combine to form a unique chromosomal set. But more on that in the next episode. So let's begin with an overview of typical reproductive physiology and function for the average male, and those whose main reproductive organs include testes and their tubules, and an epididymis, all of which are housed in the scrotal sac, or scrotum. Each testis is suspended inside the scrotal sac by a spermatic cord. Inside the spermatic cord are muscle fibers, nerves, blood vessels, and a sperm traveling tube called the vas deferens. The vas deferens begins where the epididymis ends and heads upward inside the spermatic cord and loops around the bladder to connect with some very important vesicles and glands, all of which connect to the urethra, and the urethra is the only passageway out of the body through the penis. So let's pause for a second at the scrotal sac, because this is not only where sperm begins its production, but it's also our first homeostasis stop. Sperm production, also called spermatogenesis, requires optimal temperatures that are about 2 to 4 degrees Celsius lower than core body temperature. So its set point is somewhere around 34 degrees Celsius. The scrotum has loads of thermoreceptors in its very thin layer of skin, which constantly monitor scrotal temperature. And these receptors, or sensors, send signals to the central nervous system, alerting a control center to activate an effector to regulate the temperature when it goes beyond its set point. When the scrotal environment is too cold, the brain tells the effector to contract. In this case, the effectors are the inner layer of the scrotum called the tunica dartus and the muscles within the testes. And these effectors work together to pull the testes up closer to the body to warm up. If the testes get too warm, well, surprisingly, I couldn't find a specific homeostatic feedback loop that kicks in, but rather a number of physiological factors that are sort of built in to help constantly release heat, such as the positioning of the testes away from the body. The very thin skin surrounding the scrotum helps prevent overheating. The skin also contains loads of sweat glands, which, unless the scrotum is always sweating, there should be a negative feedback loop regulating sweat initiation and cessation. There also seems to be some vascular wizardry involving countercurrent arterial cooling, which reads a bit like built-in heat pump style air conditioning, involving a very specific blood circulation mechanism. You might have to look that one up. I'm not sure I fully understand what's happening there, but I think you get the gist. There are homeostatic mechanisms in place to keep the testes at optimal sperm production temperatures. Okay, so let's take a peek inside the testes to see how and where sperm is produced and stored. So sperm production begins around the onset of puberty in tubules called the seminiferous tubules. These are thin tubes that coil into conical lobules, and there are about 250 of these coily cone lobules inside each testis. At the base of the membrane of these tubes, there are germ cells that differentiate into baby sperm cells called spermatogonia, and another type of cell called Sertoli cells. And these are very important cells because they nourish developing sperm cells. Now, sperm cells are produced in 3 stages. The first stage involves the central nervous system. At puberty, the hypothalamic pituitary gonadal axis reaches maturation. And at this point of maturity, the hypothalamus in the brain starts releasing gonadotropin-releasing hormone, or GnRH. And this signals to the anterior pituitary gland to release follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. So these hormones are being released into the blood. Now, in between the seminiferous tubules are interstitial cells called Leydig cells, which also happen to be in direct proximity to blood vessels, which is very convenient because these Leydig cells are the main production hub of androgens, including testosterone. To be clear, androgens began production in early gestation and contributed to the development and growth up until this point. But at puberty, there is a surge in luteinizing hormone hurtling through the bloodstream, signaling to the interstitial cells to produce and release a lot more testosterone. At puberty, testosterone levels spike and drive all kinds of changes in the body, one of which is waking up the reproductive system. Pause for homeostasis note. Testosterone does have its own built-in negative feedback loop. When testosterone reaches high concentrations in the blood, that signals to the hypothalamus to suppress the release of gonadotropin-releasing hormone, which puts a halt to, or significantly diminishes, the signal to the pituitary gland, which by extension diminishes the release of luteinizing hormone. And without luteinizing hormone high-fiving the Leydig cells, they won't be stimulated to produce and secrete testosterone. But as I said, at puberty, testosterone levels spike and remain fairly constant for quite some time because it's involved in a lot of pubertal business. How it wakes up sperm production is that simultaneously, inside seminiferous tubules, the Sertoli cells respond to the follicle-stimulating hormone that was released by the pituitary, and their response is to secrete a few really important molecules, and among them is androgen-binding protein. Androgen-binding protein does what it says on the tin and binds to the androgen testosterone, and it does this to sequester large local concentrations of testosterone. And when this happens, it triggers baby sperm stem cells called spermatogonia to begin the process of mitosis. Mitosis is when cells in your body divide into two completely identical versions of itself. We'll call those daughter cells. Now, technically, mitosis has already been occurring with these baby stem cells, spermatogonia. But at puberty, testosterone signals it is time for spermatogonia to divide differently, so that instead of dividing into two identical spermatogonia, the cells divide into two different spermatogonia cells. One stays back so it can divide again and keep the process going, and the other makes its way toward the center of the seminiferous tubule called the lumen. And somewhere between the basal membrane layer and the lumen, the second spermatogonium becomes a primary spermatocyte. Then, in the second stage of spermatogenesis, these primary spermatocyte cells will go through meiosis, not to be confused with mitosis. Meiosis only takes place in the production of gametes, so both sperm and oocytes go through meiosis. Meiosis, in a nutshell, is the stage of gamete production where parts of the chromosomes inside the genetic material of spermatocytes or oocytes are recombined so that every gamete has a different genetic makeup. So in sperm production, this means every sperm cell has a different genetic combination. This genetic variety is why you are different from your biological parents and your biological siblings if you have any. Meiosis is also the process by which each gamete ends up with half the number of chromosomes as the other body cells. This part of the meiosis process is called reduction division. So each primary spermatocyte and primary oocyte begins with 46 chromosomes, and a cell with 46 chromosomes is known as a diploid. But as primary spermatocytes or oocytes divide in meiosis, they only bring half the chromosomes with them into the divided cells, and a cell with half the number of chromosomes as its pre-divided original is called a haploid. Meiosis is carried out very differently in oocyte production, but we'll get into that in a minute. So at the end of one primary spermatocyte's meiotic reduction division, we have 4 round haploid cells called spermatids that now need to mature in the third stage of spermatogenesis. Important note, meiosis is continuously initiated in the testes for as long as spermatogonia are available to divide, and that can keep going on into very old age. Again, this is very different from female gametogenesis, and importantly, related to health and wellness, problems that occur during spermatogenic meiosis can lead to meiotic arrest. And in sperm production, this results in the inability to produce those all-important spermatids, or at least properly formed spermatids, necessary to mature into viable sperm cells. Meiotic arrest results in a condition called azoospermia, which could be responsible for roughly 10 to 30% of male infertility. On a more fun side note, microtubules have a very interesting role in meiosis, in which they form long spindle fibers, which they use like long arms to pull apart chromosomes during reduction division, like some kind of supervillain, except they're not at all villainous. If you don't remember some of the other amazing things microtubules can do, please revisit the episodes on cell physiology and neuron physiology. If you'd like a more in-depth description of meiosis, I've posted a couple of video links in the show notes. And if you're interested in learning more about meiotic arrest in sperm cells and its involvement in azoospermic infertility, I've posted a few studies in the references section of the show notes. They're pretty intense papers, but if you familiarize yourself a little more with meiosis and its phases from primary spermatocyte to spermatids, that might help you decode some of the academic lingo. Okay, so the third stage of spermatogenesis, or sperm production, is spermiogenesis. And this process is where the spermatids we have at the end of meiosis mature and become spermatozoa, or what we typically think of when we see a diagram of a sperm cell. This can take many weeks. The spermatid goes through its most recognizable morphological changes in spermiogenesis. There are many steps in spermiogenesis. I won't go through them all, but a quick overview of what happens at this stage is the nuclear DNA in the rounded spermatid cell greatly compacts and elongates to become the head of the spermatozoa, and it develops a cap-like organelle called the acrosome, and that will be really important to remember when we get to fertilization in the next episode. The major structural change is the beginning of a tail-like structure formed by microtubules. Importantly, it hasn't yet developed the components needed to produce energy or motility. That won't begin to happen until the emission phase of ejaculation, and we'll cover that in the next episode. But now the newly formed spermatozoa needs to make its way to the epididymis to mature. So the seminiferous tubules push the spermatozoa through its tubular labyrinth by way of peristalsis and fluid secreted by the Sertoli cells. They pass through another branch of ducts called the rete testis before they exit the testis and move into the epididymis. The epididymis is another set of coily tubes and ducts that are very long. And as the spermatozoa travel through these tubes, they will each develop a flagellum, which is a single microtubule-based cilium that extends to become the whole tail and is necessary for motility. And at the top of the flagellum, but before the head, it will recruit 50 to 75 mitochondria and densely pack them into what becomes the midpiece area. And if you remember from previous episodes, when the sperm cell is activated at the emission phase and launched into a female reproductive tract, that mitochondria generates loads of ATP, which is the fuel the sperm cell needs to travel a great distance to find and fertilize an egg, which is actually about 15 to 20 centimeters. But if we scale that sperm cell to the size of an adult, that distance has been reported as being the equivalent to swimming 5 to 10 kilometers. I've also come across equivalencies that report the distance is as much as 8,000 kilometers. Regardless, sperm cells travel a significant distance for a cell that size. Velocity is another energy requirement, and since there seems to be considerable variability in sperm head morphology, which factors into hydrodynamics, sperm cells require energy to not just make that great distance, but also to compensate for any potential loss in velocity. So that is another reason why sperm cells pack in so many mitochondria. So, once the spermatozoa have matured with their heads, acrosome, mitochondria, and flagella, they remain inactive and stored in the epididymis until that magic moment when they are ejaculated. But if many months pass without ejaculation, the spermatozoa will be phagocytosed and recycled, or flushed out as body waste. But not to worry, because remember, meiotic division is constantly happening to those spermatogonia. And don't quote me on this, but someone has worked it out that the average sperm maker can churn out 1,500 mature spermatozoa per second, which works out to millions per day. So if that's you and anyone has ever accused you of being an underachiever, you can have a chuckle on the inside because by the time they're finished saying underachiever, you've churned out thousands of sperm cells. You are very productive indeed. Okay, so the epididymis is also where most of the abnormal sperm cells will be detected and eliminated by apoptosis, which is programmed cell death, initiated by the cell itself, and then also phagocytosed and recycled by the body or flushed out as body waste. I'm not clear on the specifics of that detection process, but like any cell that is defective, either its own mitochondrial self-destruct button will kick in. If you remember from the cell physiology episode, that's what initiates apoptosis. Or possibly immune system cells called macrophages are signaled to eliminate the cell. There is some evidence of post-testicular macrophage immune response in rodent sperm, but I'm not sure about humans. I'm also not clear on the extent of the defective sperm cell detection process because sperm with incomplete DNA or denatured chromatin structures, chromatin are what make up chromosomes in the DNA, So sperm with these and other defects can still make the journey to an egg and fertilize it. However, it often results in embryonic mortality and, by extension, infertile sperm. So from this point, let's imagine that our example epididymis is storing hundreds of millions of healthy and mature spermatozoa. When ejaculation occurs, about 1/3 of the spermatozoa inventory rapidly drains from the epididymis into a long, thin tube called the vas deferens. Remember, this tube travels upward and loops around the bladder. From here, it connects to the seminal vesicle to form the ejaculatory duct. The seminal vesicles secrete fluid that provides food for the sperm, and that nutrition enables mitochondria to activate. To exit, the sperm is ejected through the urethra with other fluids. Those other fluids are an alkaline fluid from the prostate gland, which connects to the urethra, and that fluid protects the sperm from the acidic nature of the urethra, as well as the acidic vaginal environment where it's headed. And another fluid is an alkaline fluid from the Cowper's gland that offers similar protections against acidity. The sperm plus all of these fluids combined are referred to as semen or seminal fluid. And the seminal fluid is ejected through the urethra, and if reproduction is the goal, it's usually ejected into a vagina. Within a minute of expulsion, the seminal fluids pool in the anterior vagina near the cervix, and then they coagulate, temporarily restricting sperm motility. But enzymes from the prostate fluid liquefy the clot and set the sperm free. There's a reason why this happens, involving protecting and preparing the sperm cells to make it through the cervix and uterus into the fallopian tube to fertilize the egg cell. We'll visit this in more detail in the next episode. There are, of course, other ways to accomplish reproduction through artificial and medically assisted insemination and fertilization. But for clarity, over the next 2 episodes, we're going to follow sperm cells and egg cells from baby stem cells through to maturity to typical mammalian fertilization, so you can have some context as to what these cells go through to develop, and the environments they need to remain healthy, and the environments they endure during the process of reproduction. Because any deviation or interruption or deficiency in these processes could contribute to a myriad of consequences that could lead to all kinds of problematic outcomes. And importantly, those outcomes could originate from either or both partners. Infertility doesn't appear to discriminate according to body parts. Okay, now we're going to pause there for a moment and move on to the reproductive environment of the average female and those whose reproductive organs are ovaries. We'll look at how egg cells are produced, cyclically released, and either fertilized or expelled from the body. At this juncture, I highly recommend hydrating or having a snack, because for the next long while, your thinky parts will engage in a dizzying dance of details, and I warn you now that some of them may make you queasy. The female reproductive process involves multiple starts and stops, and multiple cycles and multiple hormones, inhospitable environments, ruptures, tearing, and a literal bloodbath. And that's the healthy version of this process. So buckle in. Okay, so the main organs required to grow a human are ovaries, fallopian tubes, a uterus, a cervix, and a vagina. But if you made it through the last episode on glia, you'll remember that the brilliant Dr. Barris was born with a condition called Mullerian agenesis, in which he had external female genitalia, but he did not have internal reproductive organs, including a uterus or vagina. I mention this only to emphasize that there are conditions that are probably more common than you think, where someone who is otherwise phenotypically female can be born without the reproductive parts that are necessary to grow a human being. And as mentioned earlier, gonadal agenesis and dysgenesis can arrest proper ovarian development and interfere with the ability to produce female sex hormones. So to become pregnant and sustain embryonic development and fetal maturity, you typically need a uterus. This can be yours or a surrogate. Ovaries with mature eggs are handy if you wish to be biologically connected to your progeny. Fallopian tubes are where an egg is usually fertilized. The cervix protects and keeps baby from exiting the uterus too soon. And a vagina is also handy to provide passage for sperm to fertilize an egg, but as I've mentioned, that can be done through other methods as well. A vagina also provides passage to deliver your progeny into the world if you wish to have a natural birth. But again, other methods have been safely employed for many, many decades. Technically centuries, but since the key word here is safely for both baby and mom or parent, it's probably more accurate to say decades. Simply put, advances in reproductive technology have made it possible to start or grow your family in many ways. But again, for the sake of clarity and introductory level instruction, we're going to look at a typically complete ovarian, uterine, vaginal reproductive environment. So let's take a closer look at these organs. And we'll begin with the ovaries because this is where it all kicks off. The ovaries are almond-sized organs in the upper pelvic cavity on either side of the uterus. Unlike the testes, which are purposely distanced from the body to keep sperm cells a few degrees cooler than body temperature, the ovaries are nestled in the core of the body and do not need to worry about temperature. As far as I can discern from the literature, there doesn't seem to be any ovarian thermal receptors or the need for thermal regulation. There are a few layers of tissue surrounding the ovarian cavity. The outside layer of the ovaries is the germinal epithelial layer, and the germ cells that live there will differentiate into baby egg stem cells called oogonia. The next layer inward is a layer of collagen called the tunica albuginea, and the next layer is the cortex, which houses a substructure of tissue called the ovarian stroma. This is where loads of oocytes in an arrested state of meiosis, together with some support cells, make up ovarian follicles. And those follicles live there until they are signaled to begin maturation. But more on that in a minute. The inner cavity of the ovary is called the medulla. You have many medullas in your body. The word medulla just means inner cavity, and they are usually surrounded by a cortex. So your kidney has a medulla and a cortex, so do your adrenal glands, among other organs in the human body. The ovarian medulla is composed of connective tissue with loads of blood and lymphatic vessels, as well as nerve fibers. These blood vessels are how reproductive stimulating hormones released from the pituitary in the brain reach the ovaries to launch reproductive processes. We'll also get into that in a little more detail in just a minute. So let's look more specifically at the process of oogenesis. Oogenesis is the development of the egg cell, or oocyte, from germ cell differentiation to a mostly mature oocyte ready to be fertilized. The timeline of oogenesis is wildly different from spermatogenesis. If you remember, spermatogenesis begins at puberty and is potentially very nearly lifelong. Oogenesis has multiple starts and stops, and this will become clearer as we go, but a good chunk of oogenesis is sorted before birth. But let's back up a second to much earlier in fetal gestation. Similar to spermatogenesis, ovarian germ cells differentiate into baby stem cells called oogonia, and those undergo mitosis and rapidly divide into complete diploids with 46 chromosomes. However, unlike spermatogonia, oogonia mitosis occurs in fetal ovaries between the 2nd and 7th month of gestation. There can be as many as 7 million oogonia by the 7th month, but for reasons we don't yet fully understand, after the 7th month, a large number of oogonia drastically die off. Those that remain will grow and become primary oocytes. And here's where it gets even more interesting. The primary oocytes will go through the initial phase of meiosis and duplicate their DNA, but then meiosis is purposely arrested. If you remember from a few minutes ago, meiotic arrest in spermatogenesis can lead to infertility. Not so in oogenesis, at least not at this stage. The primary oocytes paused in meiosis then get stored with some support cells called granulosa cells and are encapsulated by basal lamina to form follicles. If you remember from the last episode, the basal lamina is like the extracellular supersuit of glycoproteins and collagen and enzymes that support the cells it's attached to. Anyway, these follicles at this stage are called primordial follicles. At birth, there are somewhere between 700,000 to 1 million primordial follicles just waiting to one day mature and be released in hopes of becoming a human. But here's the kicker. Only about half of those primordial follicles will make it to puberty. During the waiting years, while the primordial follicles wait in the cortices of the ovaries, hundreds of thousands of them will die off. So at the onset of puberty, there are roughly 500,000 primordial follicles. Mind you, even though it's a tiny fraction compared to how many sperm cells will mature over a lifetime, It is still a lot because between puberty and midlife, the ovaries will typically only release 1 egg per month, which works out to only a few hundred eggs over the female reproductive lifetime. Some will die off during the maturation process, and some will never mature beyond the primordial follicle stage. So at the onset of puberty, one continuous process and two cyclical processes kick in. The continuous process is called folliculogenesis, and this involves the recruitment and maturation of a bunch of primordial follicles until one is selected for ovulation, and the time frame from recruitment to ovulation is about a year. One of the cyclical processes is called the ovarian cycle, and it starts in the last stage of folliculogenesis and includes ovulation and post-ovulation. The ovarian cycle involves the preparation of the selected follicle to release its oocyte out of the ovary and into the fallopian tube and signal the initiation of menstruation if fertilization is not achieved. Now, the other cycle is the menstrual cycle, and that involves preparing and initiating changes in the uterus to either house a fertilized mature oocyte or shed the uterine lining to expel the oocyte that wasn't fertilized after ovulation. Reparation of the uterine lining is also part of the menstrual cycle. Both cycles transpire concurrently over 28 days, give or take a few days. There is some variability when it comes to the length of these cycles, but to keep things sort of simple, we're going to use the average 28-day cycle for context. Both folliculogenesis and the ovarian cycle, as hinted, leads to ovulation, which typically occurs around the 14th day of the 28-day cycle. Ovulation that doesn't lead to pregnancy makes the menstrual cycle possible and results in menstruation, and that typically ends and begins the cycle. So the day menstruation begins is day 1 of the 28-day cycle. The very first ovarian and menstrual cycle at puberty is called menarche, and when these cycles end later in life, this is called menopause. But more on that in a minute. So let's start with folliculogenesis, because this process needs to be well underway in order for the two cycles to even happen. Folliculogenesis is the next phase of oogenesis, and it's when a group or cohort of primordial follicles are recruited from the inventory of hundreds of thousands of primordial follicles, and that cohort of primordial follicles will get a chance to mature, though only one follicle of the cohort will become that dominant follicle that will eventually release the mostly mature oocyte during ovulation. As mentioned, folliculogenesis of one cohort of follicles takes about a year from initial recruitment to ovulation, and the recruitment and maturation of the follicle cohorts is continuous from puberty to menopause. So understandably, more than one cohort of follicles is in some stage of folliculogenesis at all times, because one mature follicle ruptures and releases an oocyte every 28 days. For comparison, at puberty, one sperm cell takes just a few weeks to get from stem cell to mature cell, but once the continuous process of spermatogenesis kicks in, roughly 1,500 sperm are produced every second and accumulate so that hundreds of millions of them can be released at the same time. Conversely, one oocyte takes a whole year to grow from primordial follicle to mature enough oocyte to get released at ovulation. But again, once the process of folliculogenesis kicks in at puberty, multiple follicle cohorts are continuously in varying stages of folliculogenesis, so that one oocyte can be ready to be released every 28 days. But still, it's only one cell per month, and that cell hasn't even completed its second meiotic division when it's released, and may never do so unless it's fertilized. But more on that in a minute. So very different processes. The one similarity is that spermatogenesis and folliculogenesis are continuous processes from puberty to cessation later in life. So let's follow one cohort of primordial follicles and their development all the way from recruitment to ovulation, with the gentle reminder that many cohorts will be at varying stages of folliculogenesis at any one time at the onset of puberty. Now, this may seem like a lot of detail, but a basic understanding of follicular development not only takes us to the cellular level of this paradoxically beautiful and also slightly mean process that affects nearly half the world's population, but it also provides context to those who may be experiencing ovarian dysfunction, such as polycystic ovarian syndrome, also known as PCOS. PCOS is the result of dysregulated androgen hormone production that interferes with follicular growth. This level of detail may also be helpful for those who are receiving medically assisted fertility treatments that induce ovulation. Also, you may also just wish to understand just how busy and complex ovaries are. Okay, so back to our cohort of primordial follicles. So again, very nearly 1 year before this cohort will have narrowed down to one dominant follicle that will release an oocyte into the wild, that is the fallopian tube. This particular cohort is recruited from the entire pool of hundreds of thousands of resting primordial follicles. The mechanism responsible for cohort recruitment isn't fully understood, but it does have me curious. Why these particular primordial follicles of all the follicles that could be recruited at that time? Are they specifically selected, or is it just by chance that this grouping becomes activated? Well, what we kind of know from animal models is that recruitment involves a lot of crosstalk between intra-ovarian regulators. Intra-ovarian regulators are signaling proteins and chemical messengers, such as growth factors, cytokines, even neuropeptides that are both generated and expressed within the ovary. Neuropeptides are usually found in abundance in the nervous system, so it's a little curious that they are also produced and expressed inside the ovary. However, there is evidence that at least one group of neuropeptides may have a significant role in folliculogenesis, and it is initiated within the ovary. Anyway, these intra-ovarian regulators are very chatty. They're actually hard at work all of the time, but they have specific communication and feedback patterns during recruitment. And one intra-ovarian growth factor in particular, called growth differentiation factor 9, or GDF9, and its expression appears to have a major role in recruitment. I don't know what causes its expression for recruitment, but what I find most interesting is that it's the oocyte itself within the follicle that produces and secretes GDF9. So it might be that the oocyte, as it expresses GDF9, it's volunteering itself for recruitment, and the other regulators possibly determine its readiness or assist in its recruitment. I'm not sure, maybe both. There's a lot left to understand at this stage of folliculogenesis, but I think we can get away with saying that a few oocytes send out GDF9 signals, and the other regulators receive the call and work together through multiple signaling pathways and complex homeostatic feedback loops to initiate the next step in follicular growth. I won't get into all of the signaling pathways and complex homeostatic feedback loops because it's well beyond introductory level instruction. But if I can leave you with an image that I think might be representative of the level of molecular activity and cellular communication going on inside the ovaries and between the ovaries, the central nervous system, and the endocrine system, imagine what the buzz of communication is like in an air traffic control tower at international travel hubs like Chicago O'Hare or Dubai airports. Very chatty, loads of crosstalk. Absolutely necessary to keep everything moving in an efficient and orchestrated manner. Because remember, there are actually multiple cohorts of follicles working their way through varying stages of folliculogenesis continuously from puberty to menopause. So if you ever have the good fortune to witness planes take off and land at a very busy airport, think of the hardworking ovary. Okay, so now we have our recruited or possibly volunteer cohort of primordial follicles, and the next step in folliculogenesis is follicular growth. The intra-ovarian regulators also stimulate different stages of follicle growth and will be the primary growth source for a long time before sources from outside the ovary lend a hand. At this stage, the intra-ovarian regulators stimulate the granulosa cells that surround the primordial follicle to change shape and divide and proliferate to form multiple layers of cells around the oocyte. At the same time, a glycoprotein layer that is possibly synthesized and secreted by the oocyte forms between the oocyte and the granulosa cells. It's called the zona pellucida, and it remains with the oocyte even after ovulation and helps facilitate fertilization later if there is contact with sperm cells. The oocyte itself is also growing, and has all kinds of business kicking off inside its genome that will contribute to its growth and its communication with the other cells within the follicle. So with the growing stratified layers of granulosa cells and the growing oocyte and the addition of zona pellucida, the follicles in this cohort have shifted from primordial status to primary or preantral follicle status. Preantral will make more sense in a minute. During the preantral stage, the granulosa cells continue to divide, and as they do, they develop a follicle-stimulating hormone receptors. Also during this stage, the oocyte secretes more of that magic GDF9 to recruit support cells called theca cells, which are a kind of interstitial cell, and they form the outermost layer of the follicle. There, they differentiate into two types of theca interstitial cells with luteinizing hormone receptors. Together, theca cells work with granulosa cells to produce estrogen hormones. An important side note is that both the granulosa and theca cells are very much like other cells and that they have many of the standard components, like a nucleus and mitochondria. And that's important to know because if you remember, mitochondria contain a self-destruct button, which initiates apoptosis or programmed cell death. And this will be handy to remember when we get to later stages of follicular development, when all but one of the follicles in the cohort will degenerate. Also important to note is that similarly to what's happening at puberty in the sperm-generating counterpart's nervous system, the hypothalamic pituitary gonadal axis in females, and those with ovaries, reaches maturation. And if you remember, this is the point of maturity when the hypothalamus starts releasing gonadotropin-releasing hormone in pulses, and this signals to the anterior pituitary gland to release FSH and luteinizing hormone. Now, it's not super clear at which stage the follicle goes from intra-ovarian regulator-dependent growth to gonadotropin-dependent growth, but there is some evidence that FSH begins influencing growth during the preantral follicle stage, as the granulosa cells develop FSH receptors. And there is also evidence that it isn't until the next stage that the follicle becomes less reliant on intra-ovarian regulators and more reliant on gonadotropin hormones to facilitate growth and maturation. Either way, once the follicle has a fully formed theca interstitial cell layer, the follicle shifts from a primary or preantral follicle to a secondary follicle. And once it has formed the theca cell layer, the follicle can start producing estrogen. And to do so, it needs luteinizing hormone to stimulate the production of androgens in the theca interstitial cells. And then they secrete the androgens, and they diffuse across the basal lamina to the granulosa cells, and there it will convert to estrogen with the help of an enzyme called aromatase, which is stimulated into production by FSH. So again, theca cells have luteinizing hormone receptors which stimulate the production of androgens, and granulosa cells have FSH receptors, and FSH stimulates the production of the enzyme aromatase, which is needed to convert the androgens from the theca cells into estrogen in the granulosa cells. This is particularly noteworthy if you are someone who has or knows someone who has polycystic ovarian syndrome, or again, PCOS. PCOS is characterized by excessive production of androgens in the ovaries, and this may be due to much higher concentrations of luteinizing hormone reaching follicular thecal cells. This leads to the theca cells producing excessive amounts of androgen, while the granulosa cells aren't getting enough FSH to produce enough aromatase to convert all of those androgens into estrogen. This particular aspect of PCOS pathophysiology is possibly due to abnormally rapid pulsing of gonadotropin-releasing hormone from the hypothalamus to the anterior pituitary, causing it to release skewed ratios of LH to FSH. What results from this androgen overproduction in the follicles is that they can't grow beyond the gonadotropin-independent stage. When follicles become dependent on proper LH to FSH ratios in the antral stage, their ability to grow and mature and become viable egg cells is thwarted when those ratios are dysregulated. The follicles will remain immature and accumulate in the ovary. There are other physiological conditions that contribute to the development of PCOS, And if you're interested in learning more about it, I posted a recent review in the show notes on what is currently understood about its development. Okay, so it's probably a good time to give some timetable context. So everything that has happened to our cohort of follicles up to this point has transpired over approximately 290 days. It has grown from about 0.003 millimeters to about 0.01 millimeter. Now, at this stage, things start to accelerate because we really only have about 80-ish days to get from a less than 1 millimeter in diameter follicle to a fully grown 20 millimeter follicle at ovulation. And just to be clear, it's the follicle that's 20 millimeters in diameter, not the oocyte inside the follicle. Okay, so let's keep going. We are at the secondary follicle stage. So morphologically, we have the primary oocyte inside layers on top of layers of granulosa cells. Then we have the layer of basal lamina, and then two types of theca interstitial cells forming the outermost layer. Within the thecal layer, there has also formed a network of blood vessels that will continue to grow with the follicle. When this vasculature has developed, the granulosa cells begin to form multiple small pockets of follicular fluid, and it's possible they're drawing the fluid in from the theca cell vasculature, as well as other secreted products from the granulosa and the oocyte. These smaller pockets of fluid will coalesce into one large pocket in the center of the follicle. This bigger singular pocket of follicular fluid is called the antrum. Once the antrum is formed, the follicle shifts from secondary to antral follicle stage, also called Graafian follicles and tertiary follicles. I'll refer to it as the antral follicle because it aligns with the formation of the antrum. The antral phase is a period of accelerated growth, and that takes about 65 days. Follicle growth becomes almost entirely gonadotropin dependent, meaning it's reliant on FSH and LH to keep growing. Follicular fluid continues to accumulate, and as it accumulates, the antrum gets larger and larger, and that shifts the organization of the follicle's internal environment. Part of that reorganization is pushing the oocyte to one pole of the cell, and granulosa cells grow and arrange themselves around the oocyte, and they differentiate into two subtypes that add more gonadotropin receptors, and that seems to be important for both selecting the dominant follicle as well as mediating maturation and ovulation later. By the end of that 65-day growth spurt, the cohort of antral follicles reaches a pivotal stage of folliculogenesis. It's at this stage that one follicle will be selected to be the dominant follicle. The gonadotropin hormone FSH plays a significant role in the underlying mechanism of selection, and it does this through a negative feedback loop. That same negative feedback loop will also trigger the termination of the rest of the follicles in the cohort in a process called atresia. Here's what happens. During a particular ovarian cycle phase called the luteal phase, which occurs roughly between day 15 and 28 of the ovarian cycle, during this phase, there is an increase in FSH released into the bloodstream. This rise in FSH affects the follicles by increasing the FSH concentrations within the follicular fluid, possibly by way of their thecal vasculature. Once the fluid in one of the follicles in that cohort reaches a specific threshold concentration of FSH, it will trigger the expression of estrogen, more specifically estradiol, which is a type of estrogen hormone. Remember, estrogen hormones are produced in the granulosa cells with the help of theca cells. So the follicle with that threshold concentration of FSH expresses estradiol, and estradiol's feedback response is to now suppress the levels of FSH in the bloodstream, causing FSH to withdraw from the other follicles in the cohort. And that withdrawal of FSH from the subordinate follicles signals to the mitochondria in the granulosa cells of those follicles to initiate apoptosis. It's possible that the signal is also mediated by some of those intra-ovarian regulators expressed by the oocyte. And if that turns out to be the case, what a way to bookend the subordinate follicle lifespan. The oocyte possibly volunteered itself for recruitment, and then possibly volunteered itself for atresia. This process of selection and atresia takes about 10 days, which may seem like a long time, but if you think about how long it took for the follicle to make it that far, it's almost a blip, really. Anyhow, I should probably clarify that the dominant follicle that arises during the luteal phase of the ovarian cycle is the follicle that will reach maturity for the next ovulation, which is about 15 to 17 days after selection. Okay, so we have our selected dominant antral follicle, and it will continue to grow by accumulating more fluid and making more granulosa cells for another 10 to 12 days. The follicle will grow from approximately 2 millimeters to 16 millimeters, and as it does so, it will increase its production of estrogen. A couple of days before ovulation, the follicle's estrogen production will sharply rise and reach a peak threshold that initiates a positive feedback loop with FSH and LH over the next 2 days. If you remember from a minute ago, during the dominant follicle selection process, FSH triggered the release of estrogen, and the modest increase in estrogen became a negative feedback modulator that triggered the suppression of FSH as a way to create homeostatic equilibrium. That's a negative feedback loop. Modest concentrations of estrogen typically tells the hypothalamus to stop pumping out gonadotropin-releasing hormone. And when that happens, the anterior pituitary no longer receives the signal to release FSH and LH, thereby suppressing those hormones. But now, right before ovulation, estrogen surpasses concentrations that would normally trigger a negative feedback loop. At higher concentrations, estrogen transitions to a positive feedback modulator. So instead of signaling to the hypothalamus to stop pumping out gonadotropin-releasing hormone, it tells it to pump out more. And when it does this, FSH levels rise, but LH levels surge way above FSH levels. And this surge in LH initiates all kinds of business in the dominant follicle. It's about to all kick off in there because now we are about a day or two away from ovulation. The LH surge activates all kinds of molecular activity that triggers and facilitates pre-ovulatory maturation of the oocyte, including finally resuming meiosis. Remember, this poor oocyte has been in suspended meiosis since before birth. So it finally gets to complete its first meiotic division, and we have to go way back to the beginning of spermatogenesis to remember what that even means. It's when the diploid cell with a full set of 46 chromosomes recombines the chromosomes and divides into 2 haploid cells with only 1 copy of the chromosomes in each cell. So the LH surge triggers the oocyte membrane to break down for division. The chromosomes reassemble into different combinations, and then the oocyte divides. But hold up, it doesn't divide into 2 equal-sized cells like the sperm cells do. The first meiotic division of oocytes creates 1 large haploid oocyte and 1 small haploid polar body. The large oocyte has its 1 copy of recombined chromosomes, and it also retains most of the cytoplasm from the pre-divided oocyte. The second small polar body still contains the other copy of chromosomes, but it barely has any cytoplasm. Both are encased with zona pellucida, which doesn't seem super necessary for the polar body because it just hangs out near the oocyte until it degenerates and gets reabsorbed by the body. But maybe the zona pellucida has some function in the polar body's degeneration. I don't know. The haploid oocyte, though, moves into second meiosis phases, but stops meiosis again partway through, and it will remain in this suspended state until it is fertilized. The follicle at this stage is the pre-ovulatory follicle, and it's reached full maturation at about 20 millimeters. It's also sometimes called the mature Graafian follicle. Meanwhile, the LH surge has triggered a molecular cascade that is breaking down part of the follicle wall by way of a controlled pro-inflammatory response in some of the theca and granulosa cells. That molecular cascade is also weakening the ovary wall and forming a hole called the stigma. So when the oocyte is released, it can exit the ovary. The mechanism of follicular rupture is not completely understood. There are theories that as pro-inflammatory responses weakens, thins, and degrades a small portion of the follicle wall, smooth muscle in the thecal layer contracts, facilitating the final rupture. It's possible further smooth muscle contractions wiggle the oocyte and some of the antral fluid out of the follicle. Now, I expected the oocyte to blast out of the follicle with the rage and fervor of a warrior goddess, but a surgeon who witnessed ovulation by chance during a procedure said it took about 15 minutes for the oocyte to emerge from the ruptured follicle. I don't know if that's true for every ovulation at every stage of life, but I have to admit 15 minutes was a little anticlimactic. Anyway, when it emerges, the oocyte is still encased in the zona pellucida and has a layer of granulosa cells on top of that. Once the oocyte and its layers are freed from the follicle, it passes through the ovarian stigma into a cavity between the ovary and the fallopian tube that is filled with fluids and secretions. How it moves from there into the fallopian tube isn't fully understood either, but it's possible that the fimbriae at the top of the fallopian tube create a current that guides the oocyte into the fallopian tube, and from there the cilia that line the fallopian tube, along with peristalsis of its smooth muscle, then move the oocyte along the fallopian tube. Somewhere around mid-fallopian tube in an area called the ampulla, or the ampulla, I don't know where the emphasis is there, ampulla, is where fertilization will most likely happen. The oocyte is only viable for about 24 hours, and it takes much longer than that to travel the entire fallopian tube. So it's up to at least one of those hundreds of millions of sperm cells to make it to the fallopian tube to fertilize the oocyte. Important to note, though, both ovaries are hard at work producing follicles, with many of them at varying stages of folliculogenesis. But only one oocyte is released from one ovary per month. It is rare but possible that both ovaries release an oocyte at the same time. It's called hyperovulation. And if both ovocytes are fertilized, they will develop into fraternal twins. But we'll stop here and pick up fertilization in the next episode. For the rest of this episode, we'll look at what happens to the ruptured follicle because its job isn't quite finished yet. Plus, we'll take a quick peek at menopause and possible age-related issues with fertility. Okay, so the follicle has ruptured and the oocyte is exited, but the follicle still has work to do. Post-rupture, its structure changes. It morphs into what looks like a tiny cronut called the corpus luteum. The direct translation, I think, is yellow body, and it becomes a temporary endocrine gland that prepares the uterine lining for implantation of the fertilized oocyte. But if the oocyte isn't fertilized, it facilitates menstruation. Here's what happens. Still mediated by that LH surge, after the oocyte exits the follicle, the granulosa cell layers fold in around the antrum, which still contains some follicular fluid, as well as blood. The theca cells follow. Both the theca and granulosa cells convert to small and large luteal cells, and in so doing develop more steroid hormone-specific receptors, so they can produce and pump out mostly progesterone, and to a lesser degree, estrogen. The secretion of progesterone signals to the uterus to get ready for the implantation of a potentially fertilized egg cell. It's technically called a blastocyst by the time it reaches the uterus, if it was fertilized, because the cell division is already well underway by then. If not, it's still an oocyte. But the uterus doesn't know just yet if it's fertilized or not. So it gets to business growing and thickening a layer of the uterine lining called the endometrium and extending blood vessels into it. So it basically becomes a cushy, secretory layer of tissue that enables the post-fertilized, already cell-dividing blastocyst to adhere to the uterine wall. But if the oocyte is not fertilized, the corpus luteum stops pumping out progesterone, then degenerates and turns into scar tissue. The fall in progesterone levels cues the uterus to shed that newly cushy endometrial lining in a process called menstruation. So the lining breaks down and tears away from the uterine wall, rupturing all of those newly established connective blood vessels. Tissue and blood is expelled out through the vagina with the assistance of uterine contractions, And this continuous expulsion can take anywhere from 3 to 6 days, and it can hurt like a mother trucker. With the exception of pregnancy or any physiological disorder that interferes with menstrual function, menstruation happens every month for about 40 years, or until it ceases. And this cessation is called menopause. But here's the kicker. When it finally ceases, for some it's a relief, and for others it can be profoundly sad. It can also be simultaneously a relief and profoundly sad. So please be kind to anyone nearing menopause. If you're feeling extra kind, thoughtful notes, mixtape playlists, puppies, and chocolate are generally kindly received offerings to the exhausted ovaries and uterus. Again, the ovarian lifetime reproductive window begins to shut down around midlife, barring any physiological disorder or medical intervention that would end it before then. There is some variability to that reproductive window, but unlike their very nearly lifelong sperm-producing counterparts, the ovaries cease to release eggs usually between the ages of 50 to 55, when menopause kicks in. Menopause marks the end of reproductive viability, brought on by a decline in the production of reproductive hormones and the end of follicle inventory. And that could be age-related decline, or sometimes medically induced, where both ovaries have been removed, or a complete hysterectomy, which is the removal of ovaries and the uterus. There are stages of age-related reproductive decline before entering full menopause, as well as age-related risks of pregnancy complications, including fetal developmental issues, and those can begin as early as 35 years of age. And though both partners are susceptible to contributing to age-related risks of fetal developmental complications, the average ovary reproductive window is considerably shorter than the sperm production window by many, many years, potentially decades. Again, there is some variability there, and though sperm production can persist until very late age, other components of seminal fluid needed to fuel and protect sperm can begin to decline by as early as mid to late 40s, and age-related risk of fetal developmental issues brought on by abnormal sperm cells can emerge before the age of 60. There are other age-related constraints on reproductive viability and fetal development for both sexes. So if you produce sperm and are planning to hold off on parenthood until you're 90, or even 50s or 60s, you may wish to speak to a urologist or fertility specialist regarding fertility treatments and methods of harvesting and storing healthy sperm cells until you're ready to start a family. Likewise, there are methods of egg harvesting and storage for those who have ovaries and wish to start a family later. It's my understanding that current methods of egg harvesting are considerably more invasive and likely cost prohibitive, but potentially something to keep in mind if you're navigating fertility options. Also, age doesn't have to be the only driving factor to consider these methods. There could be health-related circumstances or eventualities that could interfere with the proper functioning of reproductive organs at any age. Speaking with a licensed medical professional could help you plan for parenthood, but if that isn't an accessible option for you, there are loads of very helpful urologists and obstetrician gynecologists, educators in all of the usual online spaces. A few I have learned from are Dr. Fenwa Milhouse, Dr. Reena Malik, Dr. Tanea Narendra, Dr. Karen Tang, Dr. Ashley Brandt, Dr. Danielle Jones, Dr. Jen Gunter, and Dr. Jennifer Lincoln. There may also be organizations in your area with free resources that assist with family planning, so that may be of help to you too. Okay, lovelies, that's all for this episode. For homework, I have posted links to videos on the process of meiosis, other animations of gametogenesis, and an article with images of real human follicle releasing an egg that was captured by chance during a surgical procedure. So you can see what a real follicle and egg looks like just after it's been squeezed out of the follicle. Thank you all for being here. Again, please check out the STEM organizations in the show notes and support them or others in any way that you are able. I have no relationship with these organizations. I'm just a fan of the work they're doing, and I hope that you will be too. If you like this episode, please click like and subscribe and the notification bell if you haven't already. And please share it with your friends, neighbors, and family, and anyone who you think would benefit from understanding their body a little better. Until next time, take care out there. It's 1 o'clock in the morning. Starting to lose feeling in my legs. And energy. Oops, just clapped. I just spat everywhere. It keeps spitting.